everyone. So today we'll be doing about replication. Okay, since I've already told you in molecular biology, okay, about central dogma in molecular biology, central dogma in molecular biology, which tells us that first DNA replicates, okay. If this is DNA, then it will replicate, okay, newly synthesized strand, okay, it will replicate. Then after replication, from these two strands, okay, from these two strands, mRNA, that is single stranded RNA, is synthesized, okay, and this process is called transcription, and then mRNA is converted into polypeptide chain, okay, proteins, by the process called translation. So at first DNA replicates, then DNA transcribe into mRNA, and mRNA get translated into protein. Okay, so now we are doing about replication first, when DNA just doubles before cell division. Okay, so replication, when we look into the replication, here it is saying, while proposing the double helical structure for DNA, Watson and Crick scientists, they had immediately proposed a scheme for replication of DNA. They told us that how DNA replicates. Okay, they proposed that scheme. This scheme, okay, the scheme suggested that the two strands would separate, okay, the two strands, these two strands, one strand and another strand. This would at first separates like this, okay, because we cannot unwind a helical structure at a single time. Okay, we cannot unwind at a single time. So these double helical structure, at first they unwind. So they separate and act as a template for synthesis of new complementary strand. So they act as a template. Template means a referral strand, okay. When new DNA should be, when new DNA strand, okay, should be there, then this strand, the parental strand, okay, the parental strand, they act as a referral strand, and that's why this parental strand, they are called template strand. Okay, that means in a template strand, if there is a nitrogenous base, then in a newly synthesized strand, okay, comes the T, because when this new DNA is synthesized, okay, the new strand and the parental strand must be, you know, able to form the helical structure again. So in order to form that stable helical structure, the nitrogenous base must be pairing with each other, okay. That's why when in this parental strand, okay, template strand, which act as a template strand or referral strand, if there is A, then in the newly synthesized chain there will be T, and if there is G, there will be C. If there is T here in the parental DNA strand, then in the newly synthesized strand there will be A, okay. Okay, complementary base pair comes together and form a helical structure DNA. Like this. So this is a parental DNA, okay, parental DNA. At first, the parental DNA unwinds this red, okay, it symbolizes parental DNA. So we cannot unwind, okay, we cannot unwind this helically coiled structure at one time because it is very, very long. Okay, if it was very, very short, then yes, of course we could have unwind this helical structure, but since this DNA is very, very long, we cannot unwind this DNA strand at one single go. So at first, okay, in one particular point it will unwind to a certain extent, okay, then complementary uh, yes, complementary nitrogenous, sorry, nucleotide chain, okay, the new nucleotide chain of DNA will be synthesized or will get polymerized, okay, and it will again further open, then again it will continue to polymerize, again it will further open, and it will again continue to, the new DNA chain will be continually uh, it will continue to polymerize up to the length of this parental DNA, okay. And when it is polymerizing, okay, it will yes, it will form helically coiled structure with that of the parental strand, one of the parental strand. See, after the completion of replication, each DNA molecule would have one parental and one newly synthesized strand. This scheme was termed as semi-conservative DNA replication. So you can see here, okay, after the new strand is synthesized, this blue color, okay, we can see here that one strand, this will again gets wind up, okay, this will again forms a helically coiled structure. So here we can see that one is newly synthesized strand and another one is parental, one of the parental strand. So this way it is replication of DNA is semi-conservative, because when this one single DNA, okay, gets into two DNA, when one single DNA forms two DNA after replication, okay, in this DNA you can see one strand will be parental and one will be the newly synthesized strand. That's why it is said here that replication is semi-conservative in nature, okay. And it is proved experimentally also, okay. It was proven by Matthew Behanson and Franklin Steele, okay. It is the experimental proof that DNA is semi-conservative. What did they do is they grew E. coli. E. coli is a bacteria in a medium, okay. They grow E. coli bacteria, that is E. coli bacterial cell, which contain DNA, isn't it? So in a medium containing N15, okay, which is the heavy isotope of nitrogen. They grow some of the E. coli bacteria in a medium which contain heavy isotope of nitrogen as the only nitrogen source for many generations. The result was that N15 was incorporated into newly synthesized DNA. So when they incorporated this E. coli bacteria into the medium that contain heavy isotope of nitrogen, okay, and when the DNA of this E. coli bacterial cell, they replicate, okay, the newly synthesized DNA strand, okay, the newly synthesized DNA strand that contained this heavy isotope of nitrogen, okay. So this heavy DNA molecule could be distinguished from the normal DNA by centrifugation in a cesium chloride density gradient, okay. Centrifugation, okay, there is a machine called centrifugation machine in which there is a test tube like structure, centrifugation tubes, okay, you can see here in the diagram. So when it is centrifuged in the centrifugation machine, okay, the DNA, they gets the heavy particles, whether it is DNA or whatever, okay, the heavy particles always settled in the bottom. The intermediate, which is not so heavy, not so light, it gets into the middle portion of the tube, okay, and if the particle is very light, it will be on the surface, near to the surface of the tube, okay. So since this DNA, it contained heavy isotope of nitrogen, because the E. coli bacteria was grown in a medium which contained heavy isotopes of nitrogen, okay. Now then they are transferred into cell into a medium with normal nitrogen, okay, and 14H4Cl. Now the heavy isotopes, the DNA which contain heavy isotopes of nitrogen, all right, they were transferred into the medium which do not contain heavy isotopes. Now what happens is, because see, now the DNA, this DNA separates, okay, when they replicates, they separate, and newly synthesized DNA strand, it is formed. So here you can see there is a red strand, there is green strand. Red strand is the strand which contain heavy isotope of nitrogen, okay, parental strand, and the green strand is a newly synthesized strand. From one DNA there is two DNA after replication, okay. So when they are again centrifuged in the centrifugation machine, the DNA was in the middle portion, okay. The DNA did not settled at the bottom, and they do not come near to the surface also, it was in the intermediate position. So that means half of the DNA, okay, half of the DNA strand, it contained heavy isotopes of nitrogen, and half of the DNA contained normal nitrogen. You can see here in the green, okay, which is newly synthesized DNA strand, it is N14, and the red one, okay, it contained N15, that is heavy isotopes of nitrogen, okay. Again after 20 minutes, again this intermediate culture, okay, it was again placed in a medium which do not contain these heavy isotopes of nitrogen. The medium contained normal nitrogen, okay. Now what happened, see, these two separates, one green, one red, okay. These two separates, one green and one red. So again newly synthesized green one again synthesize, here also one red is there, so green will synthesize, okay. In this strand, one green is parental green and one green is newly synthesized. So here one red is parental red and green is newly synthesized, okay. Similarly this DNA also, one red parental one, one green parental one, and these two green are newly synthesized. Now when these nitrogens are again centrifuged, okay, these two nitrogen, these two DNA, okay, the DNA which do not have any of the N15 heavy isotopes, this green one, okay, which do not have, there are two, isn't it, that DNA, it comes near to the surface of the tube, and the two DNA, okay, which contain half of the heavy isotopes and half normal nitrogen, they were at intermediate position. So this tells, okay, that DNA is semi-conservative. It was heavy, and when it was cultured in a normal nitrogen, okay, some of the DNA, sorry, one of the strand was heavy and one strand was normal, okay, so intermediate the weight, okay. And when it is again, when this generation is again transferred to the medium which contain normal nitrogen, they show that half of the DNA, they are very light, and half of the DNA, they are intermediate. So this experiment proves that DNA is semi-conservative in nature. So let's read. Then they transferred the cell into a medium with normal nitrogen and took samples at various time interval as the cell multiplies, okay, and extracted the DNA that remained as double-stranded helices The various samples were separated independently on CSCL gradient to measure the density of DNA. Density means whether it is heavy or light or intermediate, okay? So they just did that experiment. Thus, the DNA that was extracted from the culture one generation after the transfer from N15 to N14 medium, okay, had a hybrid, okay, because one contain heavy isotopes, one is normal. Hybrid or intermediate density, okay, intermediate density. DNA extracted from the culture after another generation was composed of equal amount of this hybrid DNA and of the light DNA. Okay, hybrid DNA and light DNA, two light DNA and two hybrid DNA. So this experiment proved that DNA is semi-conservative in nature. All right. Now talking about the machinery and the enzymes, that means how replication happens, okay, and what are the enzymes for replication. Let's see. In living cell such as E. coli, the process of replication requires a set of catalyst enzymes, okay. For replication, enzymes must be there. So the main enzyme which is involved in replication is DNA polymerase. Always remember, okay, DNA polymerase is the enzyme that that is there in replication. Now, since it uses a DNA template to catalyze the polymerization of deoxynucleotides, now what do you understand by polymerization at first? See, there is one term, polymerization. Polymerization are nothing but polymer, okay, formation of long polymers. Now polymers can be anything, okay. Poly means more than one. So there is polymer of nucleotide, okay. In DNA or RNA there is polymer of nucleotide. So polymerization means if this is DNA, it will unwind, okay, and there will be polymerization, that is formation of long chain of nucleotide. Polymerization means formation of long chain. So in order to polymerize DNA, because in replication also new DNA strand must be polymerized, replication, that process requires DNA polymerase, okay. Now this enzyme DNA polymerase, they are highly efficient enzyme as they have to catalyze, they have to initiate, they have to speed up polymerization or the formation of a large number of nucleotides in a very short time. Since DNA is very long and it will not wait for hours and hours to get polymerized, okay, within a minute, within a second, long segments of DNA are polymerized. So this enzyme must be highly efficient, very active, highly efficient, okay. Now here it is given E. coli bacteria, okay, that has only 4.6 into 10 to the power 6 base pair in a DNA. Compare it with human whose diploid content is, human being have 6.6 into 10 to the power 9 base pair, that long length of DNA, okay. It completes the process of replication within 18 minutes. Just imagine, that means how speed, okay, how efficient this DNA polymerase must be, because it has to polymerize that long chain of DNA within a minute, within like 18 minutes, okay. So that means the average rate of polymerization, that is formation of long chain, has to be approximately 2000 base pair per second. It will form a chain of, okay, chain of nucleotide sequence which contain 2000 base pair per second. Not only do these polymerase have to be fast, but they also have to catalyze the reaction with high degree of accuracy. That means if in a template strand, okay, if there is A, the newly synthesized DNA must have T opposite to A, then only it can form the bond. If there is G, this is template DNA, okay. If there is G, there must be C. If there is A, there must be T. Similarly, C, there must be G. It must be highly accurate, okay, with a high degree of accuracy. This enzyme must forward the process of this polymerization. So any mistakes during replication would result in mutation. Now mutations are nothing but changes in the nitrogenous base sequences of a DNA. If there is any kind of changes happening, okay, then we can say there is mutation in DNA, alteration of the nitrogenous bases, okay, changes in the nitrogenous bases. Okay, furthermore, energetically, replication is very expensive process. Expensive means it requires lots of energy. So deoxyribonucleoside triphosphates serve dual process. Addition to adding a substrate, they provide energy for polymerization. So the energy that is required for the polymerization of a DNA strand, okay, polymerization of DNA strand, the energy is provided by deoxyribonucleoside triphosphates, okay. Now, in addition to DNA-dependent DNA polymerases, many additional enzymes are also required to complete the process of replication with a high degree of accuracy. Not only this DNA polymerase, there are many more enzymes that are also involved in the process of replication. Okay, for long DNA molecules, since the two strands of DNA cannot be separated in its entire length, I've already told you, okay, the DNA is very lengthy, okay, very long. So at one single go, we cannot unwind this helically coiled structure. We cannot unwind at a one single go. So here it is saying, for long DNA molecule, since the two strands cannot be separated in its entire length at a time, okay, the replication occurs within a small opening of a DNA helix referred to as replication fork. So there is one particular place or a point in a DNA, okay, there is one particular point in a DNA, okay, which act as replicating or replication fork, because the shape is fork like, okay, the shape is fork like. Now the DNA dependent DNA polymerases, that means this enzyme, they catalyze polymerization only in one direction. That means formation of long chain of DNA takes place only in one direction, okay, that means 5' to 3'. Polymerization, that is formation of long chain, always takes place from 5' to 3'. That means I've already told you in the previous videos, okay, this is the two strand of DNA. One strand if contain 3', then opposite to it will have 5', okay, and this 3' in DNA ends in a 5' OH group, and this 5' OH group ends in a 3' OH group, okay, because it is anti-parallel. The two strand of DNA is anti-parallel. I've already told you in the previous videos, you can check. Okay, now here it is saying polymerization of DNA always takes place from 5' to 3'. So okay, here it is given your diagram. See, if this is two strand, okay, if this is two strand of DNA, this is called a replication fork like structure, okay, replication fork, from where the process of polymerization is initiated. Okay, and one of the strand of DNA, it is 5 dash 3 dash, okay, and opposite to that 3 dash 5 dash. Since polymerization, formation of long chain, new chain always takes place from 5 dash to 3 dash, so uh, opposite to this 3 dash group, okay, 5 dash, new DNA will polymerize continuously, okay, will polymerize continuously. But in the another strand, okay, it cannot polymerize from here, okay, and from here also it cannot polymerize, because opposite to 5 dash is 3 dash, and 3 dash to 5 dash will never occur, okay. Always polymerization happens from 5 dash to 3 dash. So when it opens, okay, from this point only, okay, from this point the DNA polymerize, and again it will open, DNA polymerizes up to the length, then again it will open up, again it will be continuous. At one strand it will be continuous, but in the another strand from where it is opened, okay, from that point again new segments of newly synthesized DNA, new segment of DNA is synthesized, okay, because polymerization always happens from 5 dash to 3 dash, okay, 3 dash to 5 dash it will never happen 3 dash to 5 dash. With the arrow you can see here, from polymerization or the formation of chain of DNA always takes place from 5 dash to 3 dash, okay. So yes, the DNA dependent DNA polymerases catalyze polymerization only in one direction, that is 5 dash to 3 dash. This creates some additional complication at the replication fork. As a result, on one strand, you can see here, okay, on one strand the template with polarity 3 dash to 5 dash, okay, the referral strand with the polarity 3 dash to 5 dash, the polymerization is continuous. While on the other, the template with polarity 5 dash to 3 dash, the polymerization is not continuous, it is discontinuous. So at last, after completing this kind of process, okay, after completing this replication process, at one strand there's nothing to do because the polymerization happened continuously, okay. But in the strand where polymerization happened in a segmental form, okay, segment, after completion of replication, these segments are joined by DNA ligase. It is joined, these fragments are later on joined by the enzyme called DNA ligase. That's why sometimes you can get question, okay, in exams, like in molecular biology, which enzyme act as a glue, molecular glue, then at that time your answer will be DNA ligase, because it will join the fragments after replication, okay. So DNA ligase acts as a molecular glue. Okay, now the DNA polymerases, the enzyme, on their own cannot initiate the process of replication. So, in order to initiate the polymerization of new DNA strand, okay, in order to initiate the process of replication, DNA polymerase cannot initiate the process by its own. So, the replication does not initiate randomly at any place in DNA. There must be proper origin of replication. There is one point called, or point or a place which is termed as origin of replication. In this origin of replication only the process of polymerization of new DNA strand are initiated, okay. So that is all about replication. So what happens at first, one parental strand, okay, DNA strand unwinds. It will not unwind at a single go, okay, because the DNA is very, very long. So it will make a shape of fork, okay, which is called as replication fork, where process of replication is initiated, or the polymerization of new DNA strand are initiated. And okay, and the DNA strand with the polarity 3' to 5', okay, the template strand of DNA, template means referral strand, okay, in that template strand of DNA, the new strand which is formed, they are continuous, because polymerization always happens from 3' to, sorry, 5' to 3'. But the template DNA where there is polarity 5' to 3', in that template, okay, the polymerization is discontinuous. So the newly strand DNA will be in a segmental form, and after the replication process is over, throughout along the length of DNA, okay, along the entire length of DNA, at last the segments of DNA are joined by DNA ligase, that is the enzyme, okay. This way at last we will get two DNA, okay, 1 and 2, two DNAs, okay, same copies of DNAs, where one strand is parental strand and one is newly synthesized strand. One strand is parental, one is newly synthesized strand. That's why it is said that replication is semi-conservative also. All right, and always remember, the enzyme that is involved in replication is DNA polymerase, okay, and replication is very energy requiring process. So the energy in the process of replication is provided by deoxyribonucleoside triphosphate. So this is all about replication.